Blood. 10 pints of life force, coursing through thousands of miles of arteries, veins, and capillaries. Those blood vessels carry blood to every other organ, keeping them functioning and thriving. Our red blood cells carry oxygen and nutrients. Our platelets stop bleeding and help to heal our wounds. Our white cells protect us against infection. And the yellow liquid plasma in which they are suspended carries an array of proteins that regulate bleeding and clotting.
Blood substitute, or also called as the artificial blood, has been synthetically developed to mimic and fulfill the function of the biological blood. It aims to reduce the chronic blood donor shortage as well as the prevalent risk of the blood-borne pathogen transmission and immune suppression during the blood transfusion process. However, what are the ethical concerns that may arise regarding the blood substitute? And what are the Islamic point of view regarding this matter?
My name is Fatih Idayu and I'll be explaining on history of blood substitute. Do you guys know that blood substitute had begun approximately 150 years ago? T. Gellert Thomas posited that intravenous infusion of cow's milk, a process termed as lacteal injection, has the potential to save lives. Despite Thomas' claim, the development of blood substitute turned to creating hemoglobin solution. Ahbs and the others performed this experiment in cats, which they completely replaced the animal's blood with a cell-free hemoglobin in a lactated ringer solution. Then, in 1950s, the US Navy treated their sailors with infusion of free hemoglobin. However, this resulted in renal toxicity problem. Several years passed, and then usable free hemoglobin solution were developed. Next, in the late 1960s, researchers named Barn and Joult cross-linked the hemoglobin with BME, which can prolong its intravascular intention. Then, using a different approach, Bonson and the others produced a hemoglobin that polymerized with glutaraldehyde, and another modification approach in which the hemoglobin is attached to a larger molecule. This is to make it stay within the system for a longer period of time.
Assalamualaikum. My name is Nurul Aina Akila binti Rosli, metric number 1716384, and now I will be explaining on the definition and types of blood substitutes. Blood substitutes are defined as solutions that are intended to replace the transfusion of banked red blood cells, but none of the products developed since then meets all blood functions. Therefore, more accurate terms for blood substitutes are volume expanders and oxygen therapeutics. There are three major classes of blood substitutes. The first one is perfluorochemicals or PFCs. These are colorless, inert and non-toxic liquids with low boiling points and are insoluble in water and alcohol. The second class is hemoglobin-based RBC substitutes or HBOCs. These hemoglobin products are different than that of whole blood, as some of them are not contained in a membrane. A few examples of HBOCs are acellular HBOCs, recombinant HB, and cellular HB. The third class is known as RBC substitutes from stem cell, and this is very useful for patients who require chronic blood transfusion or patients who have rare blood groups or autoantibodies. Despite all the developments in blood substitutes, there are still several ethical issues and Islamic points of view concerning this topic, and will be explained further in this video.
Next, we will explain about the principle and practices of blood substitute, talking mainly about the blood substitute that mimics the oxygen delivery capabilities of blood, which is called red blood cell substitute. There are many advantages for red blood cell substitute. One of them is that its ability to transport oxygen without needing of cross-match or blood typing. The second one, it can be stored in room temperature and it has a long shelf life. Also, it's free of toxicity and it has a low cost and it's very easily accessible. Also, it's fully excreted from the body and it does not accumulate in the tissues. There are two types of blood substitute. The first one is perfluorochemical based, or PFC. It's colorless and it's non-toxic. It's also insoluble in water and alcohol. That's why they use emulsifying agents to solubilize it. PFCs are capable of dissolving oxygen and carbon dioxide at a higher rate than plasma and water. However, there are many differences between PFCs and hemoglobin. The first difference, hemoglobin binds to oxygen, but PFCs dissolve oxygen. The second one, PFC is known to withstand high temperatures that reach 300 degrees Celsius, which also means that it can be easily heat sterilized. Also, the small sizes of PFCs allow it to transport in vessels easily. That's why in an in vitro study, they used PFCs as an advantageous point to reach occluded coronary arteries to maintain myocardial function.
The second type of blood substitute is hemoglobin based. The main source is human hemoglobin derived from expired red blood cell bags. Other sources include cord blood RBC, animal bovine, and recombinant hemoglobin. Animal-derived hemoglobin has some advantages, which they have unlimited access, higher resistance to degradation of heme, and the use of chloride ion instead of 2,3-DPG as an allosteric effector present in plasma. However, the transmission of infection is a very important concern in the application of hemoglobin with human or animal origin. Hemoglobin-based oxygen carriers, HbOC, are divided into acellular and cellular type. Acellular HbOC has been developed to increase hemoglobin performance and decrease its side effects. The main problem for this type is their inability to convert ferric Fe3+ ion to ferrous Fe2+ ion. As a result, methemoglobin with low oxygen carrying capacity was produced. Such complication can be avoided by attaching reducing agents to the hemoglobin surface. Next, cellular HbOC. Here, hemoglobin is encapsulated in a cell-like structure. It has the highest similarity to red blood cells, which do not cause vasoactivation due to scavenging of nitric oxide. The encapsulation of hemoglobin by a phospholipid layer, or also known as liposome encapsulated hemoglobin (LEH), may prolong its half-life and shelf life compared to acellular products. LEH is smaller than red blood cells, therefore it can pass through clots and blockage, causing more oxygenation during strokes.
Moving on to the next part, which is the ethical issue on blood substitute, there are three issues that we would like to highlight here. Firstly is the issue on how blood substitute increase the risk of heart attack by triplets. According to an article published by Oxford University 2008, a study was conducted to a total of 3,700 patients using blood substitute. The result to this study displayed that there is a 30% higher risk of death overall for patients who receive transfusion using the blood substitute. It is due to the hemoglobin in the blood substitute which scavenges nitric oxide from the blood, causing blood vessels to constrict and sticky platelets to build up, thus increase the risk of heart attack. Food and Drugs Administration (FDA) concludes that artificial blood was unsafe. However, trials on the use of blood substitutes are allowed to be conducted on the basis that artificial blood may be useful for different conditions. Secondly, the dilemma of the paramedics to enroll in the Phase 3 clinical trial, which involved life-threatening patients. The Phase 3 clinical trial is about testing the effectiveness of blood substitute to life-threatening patients such as hemorrhagic shock victims who critically need blood transfusion. The dilemma arises since it involves life of a person and it seems impossible to obtain from proper consent from the patient to get him transfused with the experimental fluid. However, the infusion of the experimental fluid might maintain the patient's vital long enough to be transported to a trauma center. Thirdly, the ethical criticism in the study design. The wave-informed consent rule stated that in order for the rule to be used, the available treatment must be unproven and unsatisfactory. However, in the study design of polyheme, the treatment used for polyheme subject has violated the study design because they continue to use polyheme even when the blood is readily available as they reach the emergency department. This is considered unethical because blood is a proven therapy and should be employed when it is available. Indeed, an effective study design is crucial because it follows scientific process and community ethical standards. All in all, being clear with the ethical issue of a certain topics allow the community to determine how much risk the population will accept to save life in short term and to reduce mortality in the future.
Moving on to the Islamic approach or perspective to this blood substitute technology, the topic that we are currently having a discussion to understand deeper about how Islamic view toward this kind of treatment. So, according to the some writers for Islamic perspective, the authors of the articles suggested that this topic is actually quite controversial to the extent that it has sparked many questions among the Muslim community, within the Muslim communities, to understand deeper about this kind of treatment. So, it is important for us to understand, firstly, seeking a treatment or cure for disease or illness is actually permissible. So, we can refer this kind of suggestion or statement from two hadith. The first one, according to the hadith narrated by Abu Darda, Allah has sent down both the cure and the disease. He has appointed a cure for every disease. So treat yourself medically and use nothing unlawful. Second hadith from the Messenger of Allah said that Allah prohibited unclean medicine. So basically, all medicines being used must be not in category of unclean. Second, containing any impurities. So in Islam, however, blood or blood derivative products are considered unclean. So the same case goes to our blood substitutes technology. However, it's important for us to understand deeper from the Islamic scholars' perspective. According to the Mufti of Federal Territories, according to some scholars, they have already written the guidelines in their books. There are two guidelines for us to understand. The first one, there must be no alternative available to the medicine as a treatment. Must be the choice of the treatment or the choice of blood substitute technology must be the only option to save the patient. Second, it is confirmed by the medical experts that the medicine derived from blood, which is the blood substitute technology, is medically effective to cure the disease. So it is important for us to understand these two guidelines to understand deeper about the blood substitute technology, and no cosmetic purposes must be used. So it is hoped that this Islamic approach to this kind of technology can help us to understand deeper about this kind of treatment.
As a conclusion, in Surah As-Sajdah verse 7, meaning He who created all things in the best way, and He began the creation of man from clay. Allah created this world under the dictate of His wisdom and under the essential of what is beneficial for the whole system. Everything has their own beauty, and the most beautiful and better than all is the making of man. Despite all technologies in the world and all human efforts in making other human that similar to their original, no one can match Allah. Among all types of substitute, there is no substitute that could replace the vital function of human blood. The substitute that been invented could only work on one specific function. Lastly, our body system is one of Allah's greatness. Always be grateful on what happened in our life every day and appreciate our own self. With that, thank you.